Good morning, everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I'm excited to be here with you. Today, we'll be exploring the principles and instrumentation behind gas chromatography, mass spectrometry, or GC-MS. Over the next few slides, I'll walk you through how this powerful analytical technique works and its applications. So let's get started and dive into the fascinating world of GC-MS. Let's dive into some advanced techniques in analytical chemistry. First up is GC-MS, also known as tandem MS. This method allows for even more precise identification and quantification by using two stages of mass spectrometry, which helps in complex sample analysis. Next is headspace GC-MS, a technique that focuses on analyzing volatile compounds that are present in the gas phase above a sample, making it ideal for flavors, fragrances, or residual solvents. Then we have pyrolysis GC-MS, which involves breaking down complex materials at high temperatures to understand their composition. Think of it as a way to analyze materials that aren't easily vaporized. Now, to give a quick overview, GC-MS, or gas chromatography mass spectrometry, is a powerful hyphenated technique. It combines two methods: gas chromatography, which separates the components of a mixture, and mass spectrometry, which helps identify and quantify those components. This combination makes GC-MS incredibly versatile. You'll find GC-MS widely used across various fields. In pharmaceuticals, it helps in drug development and quality control. In forensic science, it's essential for analyzing crime scene evidence. And in environmental monitoring, it's used to track pollutants and ensure safety. Overall, these advanced techniques build on the core capabilities of GC-MS, enhancing our ability to analyze complex samples with greater sensitivity and specificity. Let's take a closer look at the GC separation principle. Essentially, separation in gas chromatography is based on three main factors: volatility, boiling point, and how the sample interacts with the stationary phase. The idea is that different compounds will behave differently as they travel through the column, allowing us to separate them effectively. The mobile phase used here is typically an inert gas, like helium or nitrogen. These gases don't react with the sample, which helps maintain the integrity of the separation process. The output we get from this process is called the retention time, or RT, which tells us how long each compound spends in the column before reaching the detector. Now, moving on to the principles behind GC-MS. It all starts with volatilization, where the sample is converted into a gas. Then, this gaseous sample is separated within the GC column based on its properties, as we just discussed. After separation, the sample enters the mass spectrometer, where ionization occurs. Detection in the MS is based on the mass-to-charge ratio, or MZ, which helps us identify the compounds. The flow of the entire process begins with the sample, which vaporizes, then undergoes separation in the GC column, followed by ionization in the MS, and finally, mass analysis and detection. So in summary, GC-MS combines these steps: vaporization, separation, ionization, and detection, to give us detailed information about the sample's composition. Let's take a closer look at the instrumentation overview. The main components of this system include the gas chromatograph, which is essential for separating the different compounds in our sample. It acts like a sophisticated filter, allowing us to analyze complex mixtures. Next, we have the interface, which serves as the bridge connecting the chromatograph to the other instruments, ensuring smooth data transfer. Then there's the mass spectrometer, a powerful tool that identifies and quantifies the compounds after separation. It provides detailed molecular information that is crucial for our analysis. Finally, we have the data system, which collects, processes, and displays all the information, making it accessible for interpretation. Now, moving on to the principles, or as we sometimes call it, the quant part. These components work together based on fundamental scientific principles, like how the gas chromatograph separates based on volatility and affinity, and how the mass spectrometer uses ionization and mass-to-charge ratios to identify molecules. Understanding these core components and principles is vital because they form the backbone of our analytical method, enabling us to get accurate, reliable data. This overview ties into the overall theme of how sophisticated instrumentation drives precise chemical analysis. Let's take a closer look at some common sample introduction methods used in gas chromatography. First, we have split injection, which involves splitting the sample so only a portion enters the column, making it ideal for higher concentration samples. Then there's splitless injection, where the entire sample is introduced into the column, perfect for trace analysis when sensitivity is critical. Lastly, headspace sampling is a technique where we analyze the volatile compounds in the gas phase above a liquid or solid sample, which is especially useful for analyzing samples that are difficult to inject directly. Moving on to the components of a gas chromatograph, these are crucial for accurate and efficient analysis. The carrier gas system supplies the mobile phase, and commonly used gases include helium and nitrogen due to their inert properties. The injector is where the sample is vaporized, transforming from a liquid or solid into a gaseous state suitable for analysis. Next, we have the column, often a capillary type coated with a stationary phase. This stationary phase interacts with the sample components, allowing separation as the gases travel through it. The oven then plays a vital role by controlling the temperature, following a programmed temperature profile that optimizes separation and detection. Understanding these components and techniques highlights how gas chromatography is a powerful tool for trace analysis, providing high sensitivity and specificity. Each part works together to ensure precise separation and accurate identification of compounds, which is essential in many analytical applications. Let's take a closer look at the ionization techniques used in mass spectrometry. The most common method is electron ionization, or EI. This is considered a hard ionization technique because it tends to produce a lot of fragmentation of the analyte molecules. While this can be useful for structural analysis, it also means some molecular information might be lost. In contrast, chemical ionization, or CI, is a softer ionization method. It causes less fragmentation, which often allows us to see the molecular ion more clearly. This can be really helpful when identifying unknown compounds. Next, we have the interface between the gas chromatograph and the mass spectrometer, often called the GC-MS interface. Its main job is to transfer the analyte from the GC to the MS. It has to operate at high temperatures to prevent condensation of the analyte, ensuring a smooth transfer. Maintaining this temperature is crucial for efficient ion transfer and accurate results. Now let's discuss the mass analyzer itself. The most common type is the quadrupole, which is popular because of its reliability and speed. Other types include the ion trap and the time-of-flight analyzer, or TOF. These devices work by separating ions based on their mass-to-charge ratio, or MZ, which is fundamental for identifying and quantifying compounds in the sample. So, in summary, understanding these ionization techniques and mass analyzers helps us optimize our analytical method for better sensitivity, selectivity, and reliability in our analyses. Now, let's walk through the working mechanism of this analytical process. First, a sample is injected into the system, and it gets vaporized to turn into a gas. This vaporized sample is then carried along by an inert gas, usually helium or nitrogen. As it moves through the column, separation occurs based on the different properties of the compounds, allowing us to distinguish them effectively. Once separated, the compounds enter the mass spectrometer, or MS. Inside the MS, ionization takes place, typically through electron ionization or chemical ionization, which converts the molecules into charged particles. These ions then go through a process called fragment separation, where different fragments are sorted based on their mass-to-charge ratios. The detector captures these ions, and the data is analyzed to produce meaningful results. The detector, often an electron multiplier, generates signals that are processed into a chromatogram, known as TIC, or total ion chromatogram, and a mass spectrum. Each peak in the spectrum represents a different compound, with a unique fragmentation pattern that helps us identify it specifically. This entire mechanism is crucial because it allows us to analyze complex mixtures with precision and confidence. Let's talk about instrument qualification, which is a critical part of ensuring our laboratory instruments meet the necessary standards. According to USB 1058, there are 4 key stages we need to focus on: design qualification, installation qualification, operational qualification, and performance qualification. Design qualification, or DQ, is about making sure the instrument design meets our intended use and specifications from the start. Installation qualification, or IQ, verifies that the instrument has been installed correctly and according to the manufacturer's instructions. Then, operational qualification, or OQ, tests whether the instrument operates as expected under normal conditions. Finally, performance qualification, or PQ, confirms that the instrument performs reliably during routine use. All these steps are essential because they help ensure the reliability of our instruments, which is foundational for producing accurate and consistent results. When we talk about validation parameters, we're referring to specific tests like specificity, which confirms that the instrument measures what it's supposed to without interference. Linearity, ensuring the response is proportional across a range. Accuracy and precision, which verify the correctness and consistency of measurements. And limits of detection and quantification, or LOD and LOQ, which define the smallest amount we can reliably detect or quantify. Robustness, on the other hand, assesses how well the instrument performs under varied conditions. All of these elements are not just about quality, they're essential for regulatory approval. Proper qualification and validation give confidence to regulators that our instruments are reliable and that our data stands up to scrutiny. So, in summary, instrument qualification is a cornerstone of maintaining quality, compliance, and confidence in our analytical processes. Let's take a closer look at an example of residual solvent analysis using scientific methods. In this case, we're focusing on methanol as the solvent. Gas chromatography, or GC, is employed here to separate the solvent components effectively. Once separated, mass spectrometry, or MS, detects specific ions. For methanol, one key fragment identified is Mz equals 31, which corresponds to the CH3O+ ion. This type of analysis is particularly important in drug quality control, ensuring that residual solvents are within safe and acceptable limits. Moving beyond pharmaceuticals, residual solvent analysis has broader applications. In forensic science, it helps in drug identification, such as confirming narcotics. In environmental monitoring, it's used to detect pesticides and pollutants. And in the food industry, it assists in analyzing flavor and aroma compounds. So, as you can see, this analytical approach is versatile and critical across various fields, helping us ensure safety, compliance, and quality in many different products and environments. Let's talk about some of the advantages and limitations of this technique. First, on the advantages side, it offers high sensitivity, meaning it can detect even very small amounts of compounds. This is crucial in many analytical applications. It also provides excellent separation of compounds, which helps us distinguish between similar molecules more effectively. Plus, it allows for reliable compound identification through library matching, giving us confidence in our results. And importantly, it's highly reproducible, so we can trust that repeated analyses will yield consistent data. Now, on the limitations side, we need to be aware that this method is only suitable for volatile and thermally stable compounds. If the compounds aren't stable at high temperatures or aren't volatile enough, this technique won't work well. Sample preparation can also be quite involved, adding time and complexity to the process. And lastly, the instrumentation required is expensive, which can be a barrier for some labs or applications. Understanding these pros and cons helps us determine when and how to best apply this technique within our broader analytical strategies. Let's wrap up with the conclusion. When it comes to volatile analysis, GC, MS really is considered the gold standard. What makes it so powerful is that it combines two essential processes: the separation capabilities of gas chromatography, and the identification strength of mass spectrometry. This combination allows us to accurately detect and identify volatile compounds with high confidence. Because of its reliability and precision, GC-MS is widely accepted in regulatory frameworks, making it a trusted choice across various industries. You'll find it playing a critical role not only in pharmaceutical analysis, but also in forensic investigations and environmental monitoring. Its versatility and proven track record truly make GC-MS the go-to method for volatile compound analysis. For further support, please contact us. If you have any questions or need additional assistance, don't hesitate to reach out. Remember, we're here to help you succeed. Thank you so much for your time and attention today.